Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in this particular course, so far what we have discussed about the origin of life, we have discussed about the evolutions. Then subsequent to that, we have also discussed about the cellular structures. Then we talk about how the transport of the material within the cell is happening. And then we also discuss about how the cell is communicating with the another cell. And then we also discuss about the genetic material, right? So how the genetic material is varying from the prokaryotic system to the eukaryotic system and so on. Now, this genetic material is very important because it is carrying the information from the one generation to another generation, right? I'm sure you might have noticed many types of particular type of phenotypes within the human species or within the organisms. For example, you might have seen the attached earlobes. You might have seen that the thumb extends. You might have seen the headness. You might have seen the dimples, right? So you might have seen the dimples in some of the girls, right, or boys also. And then you also might have seen the color blindness also. So all these and the many more such type of phenotypes are being controlled by the genetic material what is present inside the cell. And these material are actually governing what kind of color you are going to exhibit, what kind of hair color you are going to exhibit, whether you are going to exhibit the curly hairs or straight hairs and so on. So in today's lecture, we are in this particular module, we are going to discuss about the how you can be able to study these this particular phenomena and what are the guiding principles which actually controlling the phenotypic changes into our phenotypic appearances of a particular organism. Now, before getting into this, we have to discuss about the that basic information so that you will be able to understand and you will be able to follow the content of this very complicated subject. So the basic material what is being required to carry the information from one generation to the next generation is the genetic material, and the genetic material is made up of the DNA, right? And you know that the DNA has the four different types of nucleotides. It has the adenine, guanine, cytosine, and thymine. And the information is carrying from one generation to the next generation in the form of these four nucleotides, whether it is a tiny bacteria or whether it is a big mammals or big mammals like the different types of elephants and so on, right? So all these information is encoded in the genetic material, and that's how the genetic material is going from one generation to the next generation, and that's how it is conveying that particular message to the subsequent generation. I'm sure you might have recalled that when we were discussing about the cell division, we said that cell division is also precisely controlled and precisely regulated so that there will be a complete synthesis of this particular important component of the cell, that is the genetic material, right? So once until the genetic material is not being synthesized completely, the cell will not enter into the next phase for the cell divisions. So before we get into the details, we have to understand how this particular field of science is being developed and how the people have discovered the different types of rules and laws and so some kind of things, right? So the information in the DNA, right? It determines many types of phenotypes. It determines the eye colors, right? It determines the height. It determines even the behavioral tendencies. So might have seen that some people are very short-tempered, some people are very kind, and so on. Then it also cell divides, then organ functions, or it also can actually works in the evolution also. So genetics is also been called as the blueprint of the life, which means genetics is the field of science which actually study how the information from the one generation to goes into the next generation. So genetic is the branch of biology which is concerned with the study of heredity and the variations. The term genetics was first introduced by a Batson, right, in 1906, and it is derived from the Greek word which is called as the gene, right? which means to become or to grow into. It explores how the traits and the characteristics are passed from one generation to the another through the genes and the fundamental unit of the heredity. So, gene is the fundamental unit of the heredity. I'm sure you understand more about these things when we will discuss about the central dogma and other kinds of things. The field of genetics has a profound impact on the numerous disciplines like the agricultures, medicines, evolutionary biology, and the biotechnology. Because once you know how the information goes from one generation to the next generation, you can be able to change that information, you can make it better, and that's how the people have developed different types of crop plants, different types of medicines, different types of evolutionary things, and so on. The history of genetics dated back to the ancient time when the early humans unknowingly applied the genetic principles to the selective breeding of plants and animals, which began between the 10,000 to 12,000 years back, right? So basically, even when we don't know about the law of genetics, we don't know anything about the scientific principles, people were doing the selective breeding so that they can be able to get the better breeds, better quality of the plants and the animals. Civilization like those in Mesopotamia, Egypt, China, and India selected the organism with the desirable traits to reproduce the improving crop yield and the livestock over generations. In India, the early concept of heredity was first mentioned in the ancient texts like the Ayurveda and the Manusmriti, which noted that the influence of the parental traits onto the offspring. So, and then not on the scientific methods, these observations mark the pre-scientific era of the genetics. So, this is all people were doing by simply their observational studies, like they were saying that okay, some of the bulls are very healthy, they are not getting the diseases, and they are very strong in terms of using them for the plowing and other kinds of phenotypes, and some of the cows are very good in terms of giving the milk, and that's how they were started doing the selective breedings and so on, and that's how they have actually changed the varieties. So even in the ancient Hindu text, like 2000 years back, they said that the traits are actually coming from the parents, and they advised avoiding the partners with the undesirable traits, showing the early awareness of the traits. So there were like marriages which are where you are actually avoiding one particular type of phenotypic containing parents and so on, right? Similarly, in the Jewish, they demonstrated an accurate understanding of the hemophilia inheritance, and the Greek philosophers also contributed with the theories like the pangenesis, influencing the genetic thoughts for the centuries. In modern times, the India began contributing to genetic research in the early 17th century with the pioneers like the JBS Haldane, which later moved to India and significantly influenced the genetic studies. And Indian scientists also contribute to the agricultural genetics primarily during the green revolutions, and which evolved the development of high yield crop maintaining using the genetic techniques. So before getting into the complicated subject of how the the genetics is being studied and how the genetic laws and all that, let's first discuss about the how this particular field is being evolved. You might have seen I have talked about so many different types of examples from the Hindu system or Indian old Indian system where the people were doing the selective breeding and so on. But let's take an account of how the historical aspects are being developed the field of genetics. So from the Mendel to the modern genetics, okay. So in 1856, the Gregor Mendel conducted his famous P experiment concerning the gene segregations, right? So from the 1856 to 1863, this is the time when the Mendel was doing the experiment with the P, and these are the experiments which are been the pioneers in terms of providing the different types of informations and the different types of principles on which the famous the Mendelian law of genetics were being formulated, and these are we are going to discuss in details. So in the year of 1856, the Friedrich mixture actually isolated the nuclei, the nucleic acid from the nuclei, and nuclein is also known to be DNA, right? So this was the time when the people were actually isolated the DNA, right? DNA from the particular nucleus. Then in the 1900, the Hugo de Vries, Carl Carlsons, Eric Vaughan, and all these people independently produced the result confirming the Mendel's principle of the heredity. Then 1902, the Garrod identified the first human genetic disease, and the Walter and the Boveri actually proposed the chromosomal theory of the heredity. Then 1928, the Frederick Gethe actually performed the famous experiment of transformations. So he did the experiment into the bacteria and collected the agent which is responsible for the transformation of the bacteria from one bacteria to another bacteria, and that's how you might have heard about this famous experiment where he has infected the mice with the different types of bacteria and heat killed and not killed and so on, and that's how he actually came up with the idea of transforming principles. Then in the year of 1941, the Biddle and Tatum proposed the one gene and one enzyme hypothesis. And this, according to this hypothesis, it says that the one gene is actually going to give rise to one protein. And so basically, and that's how the people have started talking about the central dogma and so on, right? And then in the year of 1952, the Hershey and Chase showed that the genetic material of the bacteriophage is DNA. And in the year of 1953, the Watson and Crick actually solved the structure of the double helical of DNA, and that's how they have actually taken out a lot of information from the DNA, and that has paved the the idea of that the DNA can be used for or be responsible for the different types of the genetic traits. Then year of 1961, the Francis Jacob and the Matthew Matheson actually discovered the messenger RNA. Then in the year of 1966, the Nirenberg and Khorana actually worked out the complete genetic codes. Then year of 1972, the Paul Berg constructed the first recombinant DNA molecule in vitro. Then year of 1977, the Walter Gilbert and the Frederick Sangers actually developed the method for the sequencing of the DNA. Then in the year of 1986, the Kerry Mullers discovered the PCR, and that's how they could be able to make the multiple copies of a particular DNA, and that's how they can be. It actually helps to generate the the multiple copies so that you can be able to use this for the subsequent recombinant DNA technology. Then year of 1997, the Roslin Institute actually clones the first cloned mammal, that is called dolly, right, from the adult organism using the technique of transgenic cloning. And in the same year, the E. coli genomic sequences was completed. Then year 1990 to 2003, the Human Genome Project completed, and that has mapped the all the genes and the genetic content of the humans, and that's how we know a lot of information about that this particular gene is controlling the skin color, this particular gene is controlling the color of the hairs and so on, right? And then the 2002 till date, there will be more advancement in terms of the genome editing techniques. So whether it is a ring finger techniques or whether it is a CRISPR-Cas, and then we also have the lot of integration of the computational tools such as AI into the genomic research. Now the question comes why we would like to study the genetics, right? We would like to study the genetics because we want to study how the different types of factors are being controlled and how these factors are going from next one generation to the next generations. So genetics is the science of heredity, right? The study of biological properties passed from the parents to the offsprings, right? And it is important that you understand the life, and if you want to understand the life, it is important that you should understand how the life is being governed, how the life process are being governed, and that's how it is important to study the genetics. So understanding the genetics is essential for explaining the inheritance and the variation among the individuals. Remember that many of these things were not known when the Lamarcks and the Darwins are actually proposed the theory of evolution, and that's how they could not be able to explain many of their hypotheses, right? Many of their phenomena, what they have observed, they could not be able to explain because the lack of the information how to explain the phenomena of zebra or the giraffes having a particular type of phenotypes. Then it actually makes the basis of genetic diseases, because if you understand the information, if you can understand the who is causing this particular type of phenotype, you can actually be able to understand the all the genetic diseases, and that's how you can actually be able to control and correct these diseases as well. Then it also helps to study the evolutions, ecology, and the population biology, and it has also been applied into the agriculture, biotechnology, and medicines. Right? We some of these examples we are going to take up by the end of this particular course, or by the end of this particular module. Then the genetic research also led to the rapid diagnostic tools, right? So you can actually be able to have the DNA-based sensors, you can actually have the DNA-based diagnostics and so on. And then it also helps in the development of the different types of pharmaceutics, like for example, we discovered the human insulin, right? So earlier, when until the recombinant DNA technology was not known, the genes for the insulin was not known, people were actually collecting the insulin from the other sources, and that's how they were actually purifying the insulin from the pigs and other kinds of animals, and that is the insulin people were using for controlling the diabetes. Then you can also have the genetically improved crops and livestock, and then you can also use this genetic for studying the forensic DNA. What are the different types of subdisciplines of the genetics? So you can actually have the transmission genetics, you can have the molecular genetics, you can have the population genetics, and then you can also have the quantitative genetics. So within the transmission genetics, it deals how the gene and the genetic traits are transmitted from generation to generation, and how the genes recombine or exchange between the chromosomes, right? So how the variations are coming from one generation to the next generation, because there is the exchange of genes between the homologous chromosomes. For example, analyzing the pattern of trait transmission in human pedigree or in the crosses of experimental organism and in examples of the transmission genetics. So transmission genetics or the classical genetics is going to allow you to study how the genetic information goes from one generation to the next generation, and in case there is a variation, how the recombinations and other kinds of phenomena are actually being responsible for making the recombination. Then we also have the another field which is called as molecular genetics. So molecular genetics is dealing with the molecular structure and functions of the genes. It is actually also dealing with the genomic analysis. Then analyzing the molecular events involved into the gene control of the cell division or the regulation of expression of all the genes in a genome. Then we also have the population genetics. So studying the heredity in a group of individuals for traits that are determined by one or only a few genes, right? So population genetics is not for studying the genetics of a single individual, but the population genetics is about the studying a particular phenomena within a particular population or particular area actually. So for example, if frequency of disease causing genes, right? So that can be very high in a particular area of within the city or within the country and so on. And that's how that kind of information you will get when you are going to study through the population genetics. Then we also have the quantitative genetics. So quantitative considers the heredity of traits in a group of individuals, but the traits of concern are determined by many genes simultaneously. For example, the fruit weight and the crop yield in agricultural plants are examples of the quantitative genetics. Now, before getting into the complicated and the details of the law of genetics, right, which is being proposed or which is being formulated by the extensive experiments done by the Mendels, it is important to study, or it is important to introduce you to the different types of terms what is going to be used in explaining those laws. So let's first understand about the different types of terms what you are going to encounter when we are going to discuss about the law. So the first is traits. Traits are characters, right? A characteristic of an individual is called as traits, right? It could be like skin color, it could be hair color, it could be eye color, it could be, so all these are actually a part of traits. A heritable trait is transmitted from generation to generation, right? This is synonyms to the character. Character in terms of the visible features, right? Not in characters in terms of the moral values or something like. It character means in terms of the features, like what is the skin color, how the what is the color of the hairs, whether the hairs are straight or curly, or whether hairs are oily, and those kind of thing, right? So examples of character include like hair colors, height, eye colors, flower colors in plants, and so on, right? Then we also have the heredity or the inheritance. The process by which the traits are passed down from the parents to their offspring is called as heredity or the inheritance. It's a reason why offspring tend to resemble their parents. The transmission of genetic information occurs during reproduction, either sexually or the asexually. Then we have the dominant traits. So traits could be of two types. It could be dominant traits or the recessive traits. So what is dominant traits? A dominant trait is one that's shown up in the organism's appearance. That is, these are the traits which are going to be visible and which are going to be appeared, right? Even if only one copy of the allele is present, okay? Even if some organism has only one gene, it is actually going to appear. It actually masks the effects of the recessive alleles, and it is representative. For the sake of argument, it is always been representative by a capital word. For example, when you are actually going to have the two different types of traits, one is dominant trait, the other is recessive traits. The dominant trait is always been written by a capital word, right? So it is going to be capital T for the tall and small t for the dog. So for example, in a TT heterozygous, the plant is tall because T tall is a dominant over the T short or the dwarf. Then we also have the recessive traits. So recessive traits only appear when the both copy of the genes are recessive, right? It is marked by the dominant alleles and only show up when the dominant allele is absent, represented by a lowercase letter, for example, the small t for the short or the dwarf. A plant is short only if its phenotype is TT, right? So recessive traits are only going to show up when they are actually going to only present. If the dominant trait is also present, then it is actually not going to be shown up. Then the third, then we have the haploids, so or the diploids, right? A cell is an individual with a one set of chromosomes, then it is going to be called as haploid. The haploid cell contains one complete set of chromosomes, right? This means there is only one copy of each chromosome, right? So for example, if humans are actually having the 46 chromosomes, right? But these 46 chromosomes are being divided into two groups like 23, 23, right? This 23 is a part of haploid. This 23 is another part of haploid. When they come together, it is going to be called as diploid. So in sexual reproducing organisms, the gametes, sperms and eggs are typically haploid. This becomes during fertilization. Two haploid gametes fuse to form a diploid zygote. So haploid is always been shown by a small n, right, and with a single n, whereas diploid is always being shown with a 2n, 2n, 3n, like that, okay. So the haploid number or the n is half the diploid number. In humans, for example, the haploid number is 23. That is what I have shown you, right? Two haploids are coming, and that's how you are actually having the 46 number of chromosomes. So a human sperms contain the 23 chromosome, and a human egg actually contains the 23 different types of chromosomes, and when they come together, it is actually going to give you a diploid zygote which actually contains the 46 chromosomes. Similarly, we have the diploids. Diploid means a diploid cell contains two complete set of chromosomes with one set inherited from the each parents. This means there are two copies of each chromosome forming the homologous pairs. Now, homologous chromosomes have the same gene in the same order but may have a different versions of those genes. The diploid number is the total number of chromosomes in a somatic cell. In humans, the diploid number is 46, which is two sets of 23 chromosomes. Then we have the DNA. So DNA we have discussed in detail, but DNA is the genetic material and it carries the information in the form of the 4 nucleotide, which is ATGC. And then we have the chromosomes. So chromosome is a long thread-like structure made up of DNA and proteins, mainly the histones, and it carries the genetic information in the form of genes. Chromosomes are found in the nucleus of the most cells, or chromosomes are present in the bacteria. Each chromosome contains many genes which determine their traits. Humans have the 46 chromosomes, 23 pairs, right? So 22 pairs of the autosome chromosomes and one pair of the sex chromosomes. So it is XX for the female and XY for the males. Then we have the gene. So what is mean by the gene, right? So a gene is a specific segment of DNA located in a particular position on a chromosome, or which is called as locus. Humans have the two copies of the each gene, one inherited from the each parent located on the homologous chromosomes. Genes are the basic unit of heredity and located on the chromosome. So every person has about 20,000 to 25,000 genes, right? Gene is responsible for eye color, right? So you can, and the gene is responsible for the height of the person. So gene is actually the main portion of the DNA which is actually going to carry the information, right? So for example, in a cell, you are going to have the nucleus. Within the nucleus, you are going to have the DNA, and DNA is going to be present in the form of chromosomes, and within the chromosomes, you are going to have the the linear DNA, and that linear DNA, a small portion of that linear DNA, which may be responsible for providing a particular type of phenotype, is being called as gene. So gene is actually the executory molecule or the smallest unit which is actually be responsible for a particular phenotype. Then we have the alleles. So alleles, an allele is a variant or the different form of gene. While everyone has the same set of genes, the alleles can be different slightly between the individual, leading to a different traits. For example, the eye color gene may have a different alleles, one for blue eyes, another for the brown eyes, right? So if you have the alleles for the blue eye, you are going to have the blue eyes. If you have the alleles for the brown eyes, you are going to have the brown eyes. It means a single gene, when it is actually going to be modified, right, there will be variations, then it is actually going to have the different types of alleles. So it can have a alleles for blue, it can be alleles for the brown, it could be alleles for the the other kinds of phenotypes, right? So while you have one gene for eye color, you may have inherit one allele from the mothers and one allele from the fathers. So that's how you can actually be able to have the different variations within the populations. Then we have the loci. So a locus is the specific physical location of a gene on a chromosome, right? So loci is kind of a address of a particular gene on the particular chromosome. So we can actually say gene X on the chromosome number 22, like that. So it is actually going to tell you about the position of a particular gene in a particular chromosome. Then we have the genotype. So the genetic makeup of an organism, the combination of alleles it carries. For example, you can have the double T, you can have the T small t, or you can have a small double t, right? All these are for the plant height, but the genotype is different, right? In this cases, you have both the chromosomes, right? You have two chromosomes, for example. So both the chromosomes are, if both the chromosomes are actually containing the capital T, then it is going to be written as capital T, capital T. If it is on one chromosome, it is capital T, the other chromosome, it is small t, then it is going to be called as T small t. And then you also going to have a third type of chromosome, where you're going to have small t and the small t on both the chromosome, and that's how it is going to be called as TT, right? So this is going to be homozygous, this is going to be homozygous, and this is going to be called as heterozygous, right? This is anyway we are going to discuss in subsequent slides. So homozygous means both the chromosomes are containing capital T or the small t, whereas heterozygous means one of the chromosome is going to contain capital T, the other one is going to be contained. So you cannot always see the genotype just by looking at which alleles are present, right? Then we have the phenotype. So genotype means genetic makeup or genetic, whereas the phenotype means the features, right? For example, you can have the brown eyes, you can have a blue eyes, and so on. So the physical expression or the observable traits resulting from the genotype, for example, the tall or the short, right? And in what you see is influenced by the other genotype and sometimes the environment, right? Then we have the homozygous and the heterozygous genotype. So homozygous means when you are actually going to have the both the genes which is present on the chromosome of same type. So if you have the alleles which are of same type, then it is going to be called as homozygous. A homozygous genotype has two identical alleles for a particular gene. For example, the homozygous dominant, so it is going to be small capital T. Homozygous recessive, then it is going to be called as small t. Then we have the heterozygous genotype. So heterozygous genotype where you have the two different types of alleles for a particular gene, but one is dominant and the one is recessive, which means on the one chromosome you are going to have the capital T, the other on is a small t. So this is going to be recessive, this is going to be dominant, and that's why this particular type of combination or this type of genotype is being called as heterozygous. For example, the TT small t, right, which carries both dominant and the recessive alleles, and the dominant allele is expressed into the phenotype. Then we have the different types of generations. So P generation, which is also going to be called as parental generations. Then we have the F1 generation, which is the first generations, and then the F2 generation, which is called as second generation. So this is the original pair of organisms involved into a genetic cross. They are usually pure breed or the homozygous for a particular trait. Example is a monohybrid cross of height in a plant like TT versus small t are the P generation. So these are actually going to be called as pure generation or pure organisms. Then we have the F1 generation. So once you breed the P generations, they are actually going to give you the offsprings, and that offspring is going to be called as F1 generation. So offspring of the P generation is going to be called as F1 generation. These individuals are usually heterozygous and show the dominant traits. For example, TT small t, when they are going to give you the offsprings, it is going to be a heterozygous T small t, which is going to be tall. Even though they carry the recessive gene, it is not expressed into the phenotype. Then we have the F2 generation. So offspring of a cross between the two F1 individual, that is been of the F2 generation. In a monohybrid cross, this generation reveals the 3:1 phenotypic ratio showing both dominant and the recessive traits. For example, if you have T small t and T small t, when they crossbreed, it is actually going to give you the 4 different types of offspring. You're going to give you the capital T, capital T, capital T small t, capital T small t, and the small t small t. This means it is going to give you 3 talls and 1 short, right? So this is going to be a part of F2. Then we have the cross, right? So we can actually have the different types of cross, which leads what are the parents or what are the plants you are going to take into the cross, that is going to decide whether it is a monohybrid cross or the dihybrid cross. A cross refers to the mating or the breeding of two individuals to show how traits are inherited. For example, crossing a tall plants with a short plant, right? It could be a monohybrid cross, it could be a dihybrid cross. So a monohybrid cross actually involves the one trait with 2 alleles, right? So it can be a one trait only, right? So monohybrid cross means it is going to deal with the one gene, which means it is actually going to talk about only one phenotype. for example, tall. So it is only going to talk about this particular type of gene, or it is going to talk about this particular type of genotype. Crossing a tall plant with a short plant, right? So it is, we are only dealing with the one phenotype, that is the height. Okay, so when you deal with the one phenotype, then it is going to be called as monohybrid cross. And when you talk about, so for example, you have the TT and small t, then it is actually going to give you the this kind of offsprings. So you are going to have a genotypic ratio like the small t, 2 small to t, and 1 small t. Whereas the phenotypic ratio is going to give you 3 tall and 1 short. Then we have the dihybrid cross. So dihybrid cross involves the two traits at the same time. So you are actually going to talk about two individuals, like for example, the tall and the color of the flower. Okay, so if you say like this, then it is actually going to be a dihybrid cross, which means if you are dealing with the two phenomena or the two phenotype, then it is going to be a dihybrid cross. For example, crossing a pea plant with the round yellow seeds with the wrinkled green seeds. So that's how it is actually going to have a dihybrid cross. So in the first generation, it is going to be show all the dominant traits, and in the F2 generation, it is going to show you the different types of phenotypic changes or different types of phenotypic organisms depending upon the different types of yields. Now, since you understood the basic phenomena, basic information about the different types of terms, what you are going to use. So let's now move on and understand the different types of law which are being formulated to understand how the genetic information is going from the one generation to the next generation, and all these laws are being formulated by the experiments what is being done onto the pea plants by the Gregor John Mendel, right? And so that's why these laws are being called as Mendel's law of inheritance. So before getting into the detail of Mendel's law of inheritance, let's first understand about the experimental design. So this is the Gregor John Mendel, right? The founder of the science of genetics. The work of Gregor John Mendel is considered the foundation of modern genetics. In the year of 1843, he was admitted to the monastery, right, in Bro, like now the Czech Republics. In the year of 1852, he began a series of breeding experiments with the green pea Pisum sativum to learn something about the mechanism of heredity, right? As a result of his creativity, Mendel discovered some fundamental principles of genetics. So from the results of crossbreeding pea plant with the different traits involving the seed shape, seed colors, and the flower, Mendel developed a simple theory to explain the transmission of hereditary traits from generation to generation. Mendel had no knowledge of mitosis and meiosis. So remember that time there was no information available about the cells, how the cell division is working and so on. And we also don't know about the genes and chromosomes and all that, right? So he did not know that the genes segregate according to the chromosomal behavior. So Mendel reported his conclusion in the year of 1856, but their significance was not fully realized until the late 1800 and early 1900. So Mendel did all his significant genetic experiment with the garden pea, which is called as Pisum sativum. The garden pea was a good choice because it fits many of the criteria that makes an organism suitable for use in genetic experiment. It is easy to grow, bear flowers and fruits in the same year, and a seed is planted and produce a large number of seeds, right? So basically why the Mendel has used the pea plant, because it is easy to breed, it is easy to and there were so many different types of traits were present in the pea plant, and it is easy to collect the seeds, it is easy to regrow and so on. So crossing the pea plant begin with a cross section of a flower showing the stamens and the pistils, right? And the pea normally reproduced by the self-fertilizations. That is, the anther of the same stamina produce the pollens, and which actually lands onto the pistil which with the same flower and fertilize the plants, right? So basically pea plant is doing the self fertilization. So within the same flower, you are actually going to have the stamen, you are going to have the pistil. So the pollen grains is going to come out from the stamen, and then it is actually going to fall onto the pistil, and that's how it is actually going to do the fertilization. Fortunately, for the success of his experiment, Mendel was able to prevent self-fertilization of the pea by removing the stamen, right? So it is actually going to remove the stamen from the few of the plants, few of the flowers, and that's how you can be able to do the crossbreeding, right? Because if it is a self-breeding, right, then it is actually, you will not have any control over how the breeding is going to work, right? So basically what he did is, he first, because the pea plants are doing the self-fertilization, he actually removed the stamens, right, which means it is actually removed the pollen tube, right? So it is actually going to, pluck off all the pollen tubes from the plants, right, so that he can actually be able to take out the pollen grains from the one plant for which he wants to do the breeding, and then he can actually be able to add those pollen tubes, pollen grains, to the desirable female plants. So next he took the pollen from the stamen of another flower and dusted them onto the pistil of the encapsulated into a pollinated. Cross fertilization, or simply cross, is the fusion of male gametes from the one individual to the female individual of another, right? Once cross fertilization has occurred, the zygote develop into the seeds, right? Certain phenotypes are analyzed by inspecting the seeds themselves, others are analyzed by examining the plant that grow from the seeds. The preliminary work ensured that the Mendel worked only for the pea strains, right, in which the traits under investigation remained unchanged from parents to offspring for many generations. Such strains are called true breeding or the pure breeding strains. So basically, Mendel has used the pea plant, and he found that the pea has actually 34 different types of strains which are pure in nature, which means they actually don't show any kind of deviations or variations. From generation to generation, they are actually showing the similar types of phenotypes, and that's how he actually used those phenotypes to understand the basic mechanism of the inheritance. So what he did, this is what he explained here, right? So what he did is, he removed the stamen, right? He removed the stamen from the, for example, he removed the stamen from this plant, right? So he removed the stamen, right? All these stamen, and then he actually took the pistil, right? So this is the pistil, right, of this particular plant, and then he taken out the pollen grains from the another plant, right? And then he did the cross fertilization, and that's how he actually produced the cross breeds. And these cross breeds, he studied whether they are showing what kind of phenotype they are actually showing. So Mendel has used the different types of traits, right? As I said, he has used the pea plant which are actually showing the true breeds, right? So he has used the different types of phenotypes. So what are the different types of phenotypes he has used? So he has used the flower and the seed coat color. So gray versus white, and purple versus white flower. Then he also used the seed color. So seed color could be yellow or could be green. Then he also changed the seed shape. So seed shape could be smooth or wrinkled. So this is what is shown here. Seed color should be yellow or green. Seed shape could be smooth or the wrinkled. Then we also have the pod color. So pod color could be green or yellow, right? So this is the pod color, green or yellow. Then we also have the pod shape. So pod shape could be inflated or pinched. So this is the pod shape, right? And then we also have the stem height. So it could be tall or short. So you can have the plants which are tall or the short. And then we also talk about the flower position. So flower position could be axial or the terminal. So this is the flower positions. You can have the axial or the terminal position. So these are the 7 different types of phenotypic combinations what he has used. And then he started studying the traits. So first what he did is, he made a simple system, right? Simple system is where you actually study the genetic movement of one particular gene, right, or one particular allele, right? So basically whenever you design these scientific experiments, right, it is important that we should first work with the simple system and then we work with the complicated system. So first thing is he studied how the genetic information of single gene moves from one generation to the next generation. So inheritance of single genes, and that's how he came up with the first law, and the first law is law of dominance. So crossing the two breeding strain bring together in the F1 one factor from each strain. The eggs, which are haploid, meaning only one set of chromosome containing one factor of one strain, and the pollen grains, which are also haploid, meaning one set of chromosome contain the one factor from the other strain. Furthermore, because one of the trait was seen in the F1 generation, the expression of the missing trait must show have been masked by the visible trait. This masking is called as the dominance, right? So for the smooth versus wrinkled, when he crossed, the F1 seeds were only smooth, right? So in the law of dominance, what he did is he did the crossbreeding of single traits between the contrasting features, right? So what he did is smooth versus wrinkled, and what he found is in the F1 generation, it is all smooth which are appeared. Thus, the alleles for the smoothness is masking or dominant to the alleles for the wrinkledness, and the smooth seed trait is considered to be the dominant traits, and the allele associated with it called the dominant alleles. Conversely, the wrinkled is recessive to smooth because of the factor for wrinkled is masked, and the wrinkled seed trait is considered to be the recessive traits, and the allele associated with it called as the recessive trait. So when the two alleles are different, one dominates and the phenotype also. So this is what is shown here, right? So if you take the two pure breeds, one is the round, the other one is wrinkled, right? So you take the seeds which are round and or the smooth versus wrinkled, right? Then in the F1 generation, it is going to only show you the seeds which are smooth, right? That says that the smooth is a dominant trait and the wrinkled is a small trait. So the genotype of the parent grown from the smooth seed is the SS, and the small s small cell is the genotype of the parent grown from the wrinkled seeds, right? An individual that contain two copy of the same allele of a particular gene are continuous to be homozygous. When the diploid plant produces the haploid gamete by meiosis, each gamete contains only one copy of the gene, which is either the so S bearing gametes, and the plant from the wrinkled is going to contain the small S bearing gametes. So when the gamete fuse, they are actually going to produce the diploid zygote, which is going to have the one small capital S and the one small s allele, and a genotype would be small s capital S. Plants that have two different alleles of a particular gene are said to be heterozygous because of the dominance of the smooth capital S allele. The small capital S small s produce the smooth seeds. So basically in a genotype, you are going to have the dominant alleles or you're going to have the recessive alleles, right? So this is going to be the capital S, which is going to be a dominant allele, right? And this is going to be small s, which is going to be a recessive alleles. And when you both of these are going to be present, then it is actually going to be the capital S is going to dominate over the small s, and that's why it is actually going to produce the the smooth seeds. So dominant and recessive alleles of a gene for the seed shape in the pea, right? So the two breeding smooth seed plant has the genotype SS, and the two breeding wrinkled seed plant has the genotype small s. Because each parent is two breeding and diploid, each must contain two copies of the same allele, and all the F1 plants produce the smooth seeds and are called as small s heterozygous. Then we have the law of segregation. So the plant grown from the F1 seeds differ from the smooth parents in that they produce equal number of two types of gametes, capital S bearing gametes and the small s bearing gametes. All possible fusion of F1 gametes are shown into the matrix called a Punnett square after its originator, Regland Punnett. These fusions give rise to a zygote that produce the F2 generation. So in the F2 generation, three types of genotypes are produced. capital S, capital S, capital S, small s, and small s, small s. So as a result of random fusion of gametes, the relative proportion of those zygote is 1 to 1 respectively, right? So however, because the S factor is dominant, so the S small s factor, both the capital S and the capital S small s seeds are smooth, and the F2 generation seeds only show a phenotypic ratio of 3:1, right? So this is what exactly it is showing here, right? So in the P generation, when you are doing the cross fertilizations, right? So you have the smooth seeds, you have the wrinkled seed when they are actually going with the fertilization in the first generation, right? So you have the capital S and capital S, small s, and that's how you are actually producing the capital S, small s, capital S, small s, capital S. So all are actually capital S and small s, and all are going to be smooth seeds because the capital S is going to be dominant over the small s, right? So basically in this one, the genotypic ratio and as well as the phenotypic ratio is going to be same, which means all are going to be smooth seeds and all are actually going to produce the heterozygous disc. Now when you take two heterozygous of these type like the capital S and the small s, and if you do the cross breeding for the capital S and the small s, when the SS is going to breed, it is going to give you the capital S, which means it is going to be smooth seeds, right? When the capital S is going to crossbreed with the small s, then also it is actually going to give you the smooth seeds because of the dominant nature of the smooth beads, right? So it is going to be heterozygous. Similarly, here also it is going to give you the smooth seeds, but in the smaller, it is going to give you the wrinkled one, right? So in the F2 generation, what you are going to have, you are going to have the genotypic ratio, which is going to be like, so if you say genotypic ratio, it is going to be like SS small, so it's going to be 1 is to 2 is to 1, whereas the phenotypic ratio, it is going to be 3:1, because still these three are actually going to show you the dominant traits, which is the smooth traits, right? So this is what is going called as the law of segregation, which means even when these two molecules or these two genes were coming together from the two individuals, in the second generation they get separated, so they are actually separable, which means the characters are not getting mixed with each other, they are pure and they will remain pure, and whenever there will be a chance of segregation, they will get segregated. So the law of segregation is said, the principle of segregation says that the recessive traits which are masked in the F1 from a cross between the two breeding strains reappear in a specific proportion into the F2 generations. So in the modern terms, this means that the two members of a gene pair segregate from each other during the formation of a gamete into the meiosis. As a result, half the gametes carry one allele and the other half carry the other allele. In other words, the gametes carry only one single type of gene from each gene. The progenies are produced by a random combination of a gamete from the two parents. Mendel also analyzed the behavior of the 6 other pairs of traits. Qualitatively and quantitatively, the same results were obtained from the 7th set of crosses. He made the following general inclusion about his data. The results of the reciprocal crosses were always the same. All the F1 progeny resembled one parent strain, indicating the dominance of one allele over the other. And in the F2 generation, the parental trait that had disappeared into the F1 generation reappeared. Furthermore, the traits seen into the F1 was always found into the F2 at about 3 times the frequency of the other traits, right? This means what it says that in the F1 generations, all dominant is going to be appear, right? Whereas in the F2 generations, the dominant and recessives are going to appear in a ratio of 3:1, whereas the genotypic ratio is going to be 1:2, 2:1. So remember that in the F1 generation, the genotypic ratio and as well as the phenotypic ratio is all going to be same, right? Because all are going to be heterozygous. In the case of here, it is going to be get segregated. To confirm this, the Mendel also has done the different types of test crosses. So at the time of Mendel's experiment, the presence of segregating factors that was responsible for the smooth and wrinkled phenotype was only a hypothesis. So Mendel was not aware of the concept of genes, right? So he could not understand why, what kind of factors are controlling and how these kind of things are happening. So to test his this factor hypothesis, Mendel allowed the F2 generation to self-pollinate. As he expected, the plant produced from the wrinkled seed are breed true, supporting his conclusion that they were pure for the small s factor, right? So he actually used the term which is called as the factor governing the wrinkleless or factor which is governing the smooth seeds. Selfing the plant derived from F2 smooth seeds produced the two different types of progeny. One third of the smooth F2 seeds were produce all smooth seeded progeny, where the other two third produce both smooth and wrinkled in each pod in a ratio of 3:1, and the same ratio was seen into the F2 progeny. These results support the principle of the gene segregation. So he actually did the test crosses. Test crosses means when you are actually breeding the offsprings together, and so when you do the self fertilization. So for example, when he did the self fertilization for the SS or the heterozygous ones, and you get the F3 progeny, it is actually going to be all smooth, right? Because all these two are true breeding. Whereas in this case, it is actually going to show you the pure population of 3:4 for the smooth or 1:4 of the wrinkled. So the capital S and the small s plants are having a different phenotype but the same dominant phenotype. The self-fertilization test, the F2 progeny proved a useful parameter to determine whether a plant with the dominant phenotype was heterozygous or the homogeneous. So test cross is a very good method by which you can be able to test, or when you can be able to say whether you are actually having a pure breed or whether you are actually going to have the crossbreeds. Because if it is a pure breed, then it is actually going to give you only that phenotype. If it is a crossbreed, then it is actually going to show you a phenotype into 3:1 segregation, which means it is actually going to show you the dominant traits of three plants and the recessive traits like. So if it is a capital N and small s, it is actually going to show you the the plants of the both the type of seeds. But if it is a small s, then it is actually going to show you only the wrinkled seeds. A common test to do this perform is a test cross, a cross of an individual expressing the dominant phenotype with a homozygous recessive plant to determine its genotype. In sum, the test crosses of the F2 progeny from the Mendel's crosses are shown to the dominant phenotype resulting into 1:2 ratio of homozygous dominant heterozygous genotype in the F2 progeny. That is, when you cross the homozygous recessive, one third of the F2 progeny with the dominant phenotype gave rise to a progeny with the dominant phenotype and were therefore heterozygous for the dominant alleles. The other two-thirds of the F2 progeny with the dominant phenotype produced the progeny with the 1:1 ratio of dominant phenotype and the recessive phenotype, and therefore with the heterozygous. But there are exceptions. There are exceptions which does not follow such kind of rules of dominant traits, right? Where the dominant traits are also, get mixed with the recessive traits. So Mendel's principles apply only to the diploid eukaryotic chromosome and forms the foundation for predicting the outcome of crosses in which the segregation and the independent assortment occurs. Whereas all the allelic pairs Mendel studied showed the complete dominance or complete recessiveness relationship. Many allelic pairs do not, as more as geneticists also did experiment through they found that the Mendel's principle did not apply exactly. So you have the complete dominance, right? Is the phenomena in which the one allele is dominant to another, so that the phenomena of heterozygosity is the same as that of the homozygous and dominant. With complete recessiveness, the recessive allele is phenotypically expressed only when it is a homozygous. Complete dominance and complete recessive are the two extremes of a range of a dominant relationship for the two alleles. But in between, you also have the incomplete dominance, right? So when the one allele of a gene is not completely dominant to another alleles of the same gene, it is said to be show the incomplete dominance, also referred to as semi-dominance or the partial dominance. With incomplete dominance, the phenotype of the heterozygote lies in the range between the phenotype of the individual that are homozygous for the either alleles. For example, if you have red and if you have white, right? So if you crossbreed, right, then it ideally it should show you the red because red is dominant over white. But what happens? Because of the incomplete dominance, it is actually going to show you a mixing of both the phenotypes, and it is actually going to give you the pink color. So the phenotype of the heterozygosity is typically referred to an intermediate phenotype through which it may not be exactly in the middle between the phenotype of two heterozygous. So incomplete dominance is also exhibited into the fruit color of eggplant. When a homozygous plant that produces the purple fruit is crossbreed with a homozygous plant with the white fruit, the heterozygous F1 generation produce the violet fruits, right? So this is actually a very simple example where these two species are two breeds, right? Two breeds for purple and white, and when they fuse, right, it is actually going to give you a violet because violet is the mixture of these two, right? So it is not showing the purple, it is not showing white, it is showing the violet. So when the F1s are crossbreed with each other, 1/4 of the F2 are purple, the 1/2 are violet, and 1/4 are white. So this 1:1 ratio is different from the 3:1 ratio that we would observe if eggplant fruit in exhibited the dominance. So there is a simple and simple differences, right? If it is a complete dominance, then it is actually going to show you a ratio of 3:1. If it is a complete independence or incomplete dominance, then it is actually going to show you a phenomena of 1:1, right? Whereas 1 is actually going to be red, this one is going to be white, and this one is going to be pink, right? So it is actually going to show you a incomplete dominance wherever you have the heterozygous alleles. So when a trait display incomplete dominance, the genotypic ratios and the phenotypic ratio of the offsprings are same because each genotype has its own phenotype. So remember that if there is an incomplete dominance, then F1 generation or F2 generation, both places you are actually going to have the similar ratio of genotypic and the phenotypic ratios. It is impossible to maintain the eggplant that are pure breeds for the violet fruits because all plants with the violet fruits are heterozygous. Some human disease show incomplete dominance. One example is sickle cell anemia, in which the homozygotes for the sickle cell mutant alleles have the sickle cell anemia, while the heterozygous that have a milder sickle cell traits. So heterozygotes should show very strong alleles, right? Strong anemia, because the sickle cell anemia is going to show up for the homozygotes, right? So incomplete dominance is exhibited when the heterozygotes has a phenotypic intermediate between the phenotype of the two homozygotes. When a trait exhibits incomplete dominance, the cross between the two heterozygotes produce a 1:1 phenotypic ratio into the progenome. And this is what is shown here in this particular figure. So you have the purple fruits, you have the white fruits, when you and these are the P generation, right? So when you do the first generation fertilization, then it is actually going to generate the violet color fruits. So it is not going to generate the purple fruits, not going to generate the white fruits. Instead, all these heterozygotes are going to generate the violet fruits. And then in the F2 generation, when these are going to crossbreed, it is actually going to give you the all sort of the genotypes, like for example capital P or small p, and then you also have a capital P and a small p. So capital P is going to give you the purple color fruits, small p is going to give you the white color fruits, and then this one is actually going to give you the violet color fruits. So it is actually going to give you the fruits in the ratio of 1:1, right? So this is the 1, this is 1, and this is going to be 2, right? So this is going to be 1 plus 1, so it is going to be 2, right? And this is all happening because of the incomplete dominance. And then we also have the codominance, right? So codominance, another modification of the dominance relationship is the codominance. So in the codominance, the heterozygotes exhibit the phenomena of both homozygotes. This means the phenotype of the heterozygote is not intermediate between the phenotype of the homozygotes. Rather, the heterozygotes simultaneously express the phenotype of both homozygotes. By contrast, incomplete dominance, the heterozygotes exhibit a phenotype intermediate between the two homozygotes. The ABO blood group system is a classical example of codominance where both the alleles contribute equally to the phenotype in a heterozygous individual. So you remember that the blood group, right? Blood group is having the two different types of alleles. You have the A alleles, you have the B alleles. So if you don't have any of these, if you have both, then it is actually going to give you AB. If you don't have any of these, then it is actually going to give you O blood group. So that's how you are going to have the 4 different types of blood groups. So if you have A alleles, it's going to be A blood group. If you have B alleles, it's going to be B blood group. If it is a both, then it is going to be AB, and if you don't have any of these, then it's going to be O blood group. So this is the phenotype, right? This is the O blood group, A blood group, B blood group, and AB blood group, right? And these are the genotype. Genotype is I by I, IA by IA, or IA IB. So this is actually in absence of the A or B genotypes. In the A, you're going to have the A phenotype, B is your B, and then AB is actually going to have the mixture of A and B. But in this one, both the A and B both are actually going to express and will give you the hybrid phenotypes. So the gene responsible for determining the blood group has 3 alleles: IA, IB, and small I, right? So if you have IA, it is going to be A blood group. If you have IB, it is going to be a B blood group. If you have neither of these but II, then it is going to be O blood group. And if you have IA and IB both, then it is going to be AB blood group. So individual with the genotype of IA or IAI, then it has a blood group of A. Those with the IB and IB or the IB and I will have blood group B, and individual with the genotype of II will have blood group O, which lacks both A and B antigen. In contrast, the I allele is recessive to both IA and IB, right? So in this kind of codominance, right, the A antigen is also being produced from the IA, and B antigen is also being produced from IB, and that's how both of the antigens are being expressed onto the RBC, and that's how it is being not showing or not following the Mendelian genetics. So IA and IB alleles are codominant, means the individual with the genotype like IA and IB expresses both A and B antigen on the surface of their red color cells, resulting into the AB blood group. This inheritance pattern demonstrated how codominance allows both alleles in a heterozygote to be fully and simultaneously expressed. So this is just a checkerboard analysis. It shows that how the offsprings are going to work and so on. Then we also have another examples of the pleiotropy. So pleiotropy, as the name suggests, right, when the 2 or more phenotypes are correlated, the traits do not vary independently. For example, the fair skin, blonde hair, and the blue eyes are often found together in the same individual. The association is not perfect. Sometimes we see individual with the dark hair, fair skin, and the blue eyes, but the traits are found together with enough regularity for us to say that they are correlated. The phenotypic correlation between the two quantitative traits can be computed by measuring the two phenotypes on a number of individuals and then calculating a correlation coefficient for the two traits. One reason for a phenotypic correlation among traits is pleiotropy, where the multiple phenotypic effect results from a single locus determining the traits. Genes rarely affect only a single trait, and this is particularly true for the polygenes that influence the continuous traits. For example, in humans, many body structures respond to the growth hormone, and there are genes that affect the amount of growth hormone secreted by the pituitary gland. People with a certain genes produce the high level of growth hormone, which increases both height and the hand size. The other processes gene develop a lower level of growth hormone, which leads to the both short stature and the small hands. So height and hand size are therefore phenotypically correlated in humans, and this correlation is due to the genetic correlation and the fact that both characteristics are affected by the same gene that controls the amount of the growth hormones. Genetically speaking, height and hand size are same characteristic because they are the phenotypic manifestation of a single set of genes. So when the two characteristics are influenced by the same gene, then they are genetically correlated. So far what we have discussed about the briefly we discussed about the how the genetics is being evolved as a field, right? So we discussed about how the people have discovered the DNA, how the Gregor John Mendel has performed the different types of experiment with the pea plant, why he has chosen the pea plant and so on. And then based on these experiments, the Gregor John Mendel has formulated the different types of the law of inheritance. So we discussed about the first law, which is the law of dominance. And then subsequent to that, we have also discussed about the exceptions to the law of dominance. So we discussed about the incomplete dominance, we discussed about the codominance, and then we also discussed about the pleiotropy. So with this brief discussion about the genetics, we would like to conclude our lecture here. In a subsequent lecture, we are going to discuss some more aspects related to the law of inheritance. Thank you.